Good afternoon, everyone. It's a great pleasure to welcome you to the first Wednesday afternoon lecture of this particular academic season. And it's great to see so many people here in Missouri, and I know there are many others watching by video, and we welcome you as well. We do this most Wednesdays. We bring an amazing set of lecturers who come here to tell us their latest results. Next week, by the way, will be Carl June, one of the leaders of cancer immunotherapy from University of Pennsylvania. So I'm sure you want to come and hear about that as well. It's a personal pleasure for me to be able to introduce today's speaker, David Reich, whose work in the area of understanding human origins is at the very leading edge of questions that all of us have asked for centuries, millennia, about where we came from and how we're all related. And using the tools of modern genomics and some pretty good sleuthing and epidemiology and figuring out how to derive DNA from ancient bones, David has been successively over the space of several years adding more and more details to that map of information about our origins and how all of us, originally Africans, have ended up in other parts of the world, and how the study of DNA can teach us about those historical events in ways that have really remarkable detail attached to them. It's also a particular pleasure to have David here as our speaker because it's kind of a family thing. Many of you know David's brother, Danny Reich, who's sitting there in the blue shirt, one of our distinguished principal investigators in the neuroimaging arena, and somebody that we have asked many times to talk about his research to distinguished visitors at various moments, including high-ranking officials of the United States government, but who does wonderful research in the area of multiple sclerosis and other things. And I'm also very happy to point out that Danny and David's parents are here also, sitting on either side of him. And his dad was once here at NIH as an investigator in NIMH. So this is really a wonderful family occasion that we have here. Well, to say very briefly a couple words about David so I don't take any more of his time. He is currently professor of genetics at Harvard. He's an associate member of the Broad Institute. He's an investigator in the Howard Hughes Medical Institute. He did his undergraduate work at Harvard in physics, always a good start, I would say, for somebody who's going to be pretty darn quantitative, and he is, and pretty mathematical as well. Then after that, did a DPhil in statistical genetics at the University of Oxford, working with David Goldstein, and then on to Harvard Medical School and MIT, but ending up at the Whitehead for a postdoc in medical genetics, and then to Harvard and the appointments that I just mentioned at the Broad and with Howard Hughes. His work has been recognized by a variety of special awards, the Newcomb Cleveland Prize from AAAS for the Best Paper in Science of the Year in 2010, the Genzyme Outstanding Achievement Award, named as one of Nature's 10 People Who Matter this year by Nature magazine last year. Happy to claim him as a PI and co-PI on several grants from NIH. And as you will hear, happy to see the way in which his research is evolving in fascinating ways, including, by the way, that his paper in Nature came off embargo at 1 o'clock today. So there's a very good bit of timing to have him here to talk about the latest findings. So please join me in welcoming Dr. David Reich.
Do people hear me? Okay, thank you. It's really a great honor to come speak here and to be invited to give this lecture to the NIH. I'm going to tell you about a new scientific instrument that's been developed in the last really 5 or 6 years, and that's really made it possible to study things that were not possible to study before. And I'm going to tell you about work that I've been involved in trying to use this scientific instrument to learn new things. So I hope that at the end of this lecture, you will appreciate the power of this new tool and its potential to learn a lot of new things. So I'm first going to tell you about this new scientific instrument, ancient DNA. So this is an instrument that was not invented on this continent. It was really developed almost entirely in Europe by European laboratories, especially Svante Pääbo's laboratory in Leipzig in Germany. But now has become a technology that other groups can use as well because of the work done there and elsewhere. So it starts with a bone that in this case, a human. I work on human bones mostly, but other laboratories work on other types of skeletal remains. We take the bone into a clean room, which is a controlled environment where there's positive pressure, so that the DNA from the outside the clean room doesn't get inside the clean room. The danger is contamination of these samples which have so little DNA in the first place. There is UV light when people aren't there to cross-link the DNA so that you don't sequence DNA that's from the researchers in the room. People wear bodysuits, and there's a lot of cleaning with bleach and use of hoods in order to clean the samples. One drills beneath the surface of the bone with a little borehole to try to get beyond where it might have been contaminated by archaeologists or people who've handled the bone. Obtains a bit of powder. That powder is demineralized and the proteins are removed through a series of chemical steps and then turned into sequenceable form and sequenced on one of the modern sequencers to produce a lot of data. So that's the basic set of steps that are used to get ancient DNA out of samples, and there's a whole bunch of optimizations that have been implemented in order to make this more efficient and really have increased the amount of DNA that one can get out of a sample by literally 10,000 times over the last approximately 6 years. So I'm just going to start with a picture of Moore's law. So Moore's law is about computer worlds, and it's where the number of elements on integrated circuits doubles every 1 to 2 years, and that's been going on for the last century, and it's driven the computer revolution. So there is a Moore's law of ancient DNA, but the rise has been much faster, and it's been much more recent. So in 2010, and I'm telling you here about whole genome data, not just mitochondrial DNA sequences, but just whole genome data where there's enough data to really learn about population variation. So if you use as a cutoff an amount where you can begin to learn about population relationships, there were 4 sequences published in 2010. There were a burst of 20 or 30 sequences published in 2014, and then it went into hyperdrive in 2015 with several hundred sequences, and it's increasing and increasing. Now we have more than 1,000 sequences in our laboratory, and I'm sure that's true also of other laboratories. And there's so much data being produced right now that there's much more data that's unpublished than that's published, just because there's not enough time to publish it. So what we've done in our laboratory, I guess it's a very American thing to do, is to make these beautiful techniques developed in Europe, beautiful piecework techniques, to make them industrial. And so we tried to take the old approach, which is still a wonderful approach of searching for a golden sample, screening and screening and screening these skeletal remains for some sample that will have enough DNA that it can be sequenced, and then working very hard on it, to a process where we screen many, many samples and and take advantage of efficiencies and techniques that makes it possible to convert a higher fraction of samples to working genome-wide data that provide the information we need. So our goal was to implement a screening process that would turn a large fraction of the samples we analyze into working data and to have a reasonable cost per sample to produce genome-wide data, in our case less than $500 a sample. One of the reasons ancient DNA is so expensive and continues to be so expensive in most implementations is that because so little ancient DNA is human. Bones are mostly, the DNA you get out of bones is mostly microbial. It's from the bacteria and fungi that ate you when you died. And so very little of the DNA that you sequence is from the individual you want to sequence, even if it's an uncontaminated sample. Beyond that, of course, most of the DNA that you sequence, even from the human, is not from the sections of the genome you're probably going to want to analyze, perhaps regions that are of interest for adaptation or for studying population relationships. So the solution that we took was a solution which is an adaptation of a method developed in Leipzig by Xiaomei Fu and Matthias Meyer. And what we did is we adapted this to a set of more than 1 million positions in the genome. And what we do is we take the DNA sample made into sequenceable form, a DNA sequencing library, and we take a technique that's used really in exome sequencing. We wash it over a series of bait sequences, typically 50 DNA letter long sequences that target the positions of the genome we're interested in. And what's left is mostly sequences that are from humans, and not only from human, but from the regions we're interested in. So most of what we're left with is human from the regions we're interested in, and then it's not a lot of sequencing to obtain data from these approximately 1 million positions in the genome. So we're currently screening and screening more than 1,000 samples per year in our laboratory, which is a huge change compared to the situation in the past. We're not the only laboratory that's able to screen substantial numbers of samples, but it's really transformative in terms of what it allows us to do. So this allows us to reduce costs and to improve quality. So here's a really wonderful sample that was published last year by a group in Denmark, which took about 100 samples and sequenced them brute force by just running the samples through the sequencer. This is the number of sequences in hundreds of thousands, and approximately the same as cost in dollars. And this was a $1 million experiment where, on average, about 20% of positions in the genome were covered. But when we apply this approach, we have many fewer sequences required and a higher average coverage of the positions that we think that are most interesting to analyze. And so it allows a great amount of efficiency and makes it economical to do this kind of work. So I'm going to tell you in the second part of my talk about a specific example about how ancient DNA has provided new insights about the past. And I'm going to talk about Europe and about Western Eurasia more broadly, not because these are particularly important places of the world, but it's because where we know the most. So this technology of ancient DNA was invented in Europe. The skeletal remains from Europe are the ones that have been surveyed first. And because of that, we have much more data. About 90% of the genome-wide data in the literature is from Europe or from Western Eurasia. And just as interesting things can be done in the Americas, in East Asia, in South Asia, in Africa, but I'm going to tell you about Europe to illustrate the power of these approaches. So the background for this is just about start with present-day data, and I want to frame this in the following way. So West Eurasia, West Eurasians, by which I mean a region that extends from Europe, on the Atlantic coast of Europe, all the way to Iran into Central Asia, in the middle of Asia, are genetically much of a muchness. They're genetically very similar. So if you measure the square of the frequency difference, that genetic variance between people across the whole regions, it's not very different. And this is actually what gave rise in the 18th century to scientific classifications of people into different groups, this sort of observation that people are genetically broadly similar at some gross level across this region. And if you look at some genetic variants, you can see this. So this is a pie chart of the frequency of variants that some people have one type and some people have another type. These are very unusual variants that are very different in frequency across populations. But you see West Eurasia clustering here with all these red dots over here. And in East Asians, from all the way from Korea to Indonesia, again, there's genetic high similarity and then a big sharp division moving into Western Eurasia. And in much of Africa, but not all of Africa, again, a lot of similarity. And the question is, how did this homogeneity across these regions arise? I think people, centuries ago, when people first began trying to make these categories, thought these are age-old things. These have existed for 40,000 years or for tens of thousands of years since populations separated from each other, or even longer, maybe it always existed. And in fact, the answer is these are only 5,000 years old, these clusters that we see today. And from ancient DNA, we know this, and I'm going to tell you how we know this in the case of Europe. So I'm going to first tell you about Europe specifically, and I'm going to tell you how Europeans today are the result of massive mixture of three highly different populations that are as different from each other as European and East Asians. It all happened in the last 9,000 years. So I'm just going to first introduce a few pieces of evidence from outside genetics, the evidence of archaeology and the evidence of language. So what you need to know about the archaeology of Europe, there's many things, but one very important thing is the arrival of farming in Europe. So farming was not invented in Europe. It was invented in the Near East, beginning between 12,000 and 11,000 years ago, probably in present-day Turkey or Syria or Iraq. And it spread from there, reaching Greece, present-day Greece, about 3,000 years later, and then it reached Britain and Scandinavia by about 6,000 years ago. It spread across Europe very dramatically. Population densities increased. Lifestyles completely changed. The types of food people ate completely changed. This was a dramatic change. The question has always been whether this massive transformation economically was accompanied by movements of people. There was a lot of skepticism that there might be such big movements of people, but it was always a question about whether it was movements of people or just adaptation of ideas. You know, we all use cell phones right now, but we're not all ethnically, genetically the same, and we adapt ideas from other people, so maybe farming was adopted from people's neighbors, or did it spread through people? The other observation is languages. So people in Europe today, almost all with exceptions like Basque and Hungarian and Estonian and Finnish, speak languages that are closely related to each other and related also to the languages of northern India, of Iran and Armenia, called Indo-European languages, with a great gap in between in the Near East, and we know it's been a gap where Indo-European languages have barely been spoken for the last 5,000 years, because writing was invented in the Near East, and we know for the last 5,000 years that except for Turkey, present-day Turkey, sometimes there haven't been these languages in this region. So what does this how does this correspond to movements of people? So in 2009, and then with genome-wide data in 2012, it became absolutely clear from genome-wide data that the arrival of farmers in Europe was accompanied by huge movements of people. And the way this was known is that people successfully got mitochondrial DNA. This is the DNA in these energy factories of your cell. It's about 15,000 DNA letters long. It's only about a 200,000th of the total length of your genome, but it was the part of the DNA that first people began to study with ancient DNA, because there's about 1,000 times more copy per cell, and the challenge in DNA is to get anything at all from these bones, and so people started with what there was the most of. So when people studied DNA sequences from hunter-gatherers from Europe, from 8,000 years or 9,000 or 10,000 years ago, what they found was all of the mitochondrial sequences were the great majority were of two types, what they called U4 and U5, defined by the mutations characteristic of those types. But the first farmers had almost no U4, U5, clearly evident of people coming in from elsewhere. Genome-wide data from people from 5,000 years ago in Sweden, which was substantially after the first arrival of farming, led by Pontus Skoglund and colleagues, then showed that already 5,000 years ago, the first farmers of northern Europe and Scandinavia were genetically very different from the hunter-gatherers who were living side by side with them at the same time. Again, about as different from each other as Europeans and East Asians, and that, in fact, were genetically very similar, not to present-day people from Sweden, but to present-day people from Sardinia. And they had a model where people in Europe today are a mixture of two ancestral populations, the farmers, who have left the most ancestry in people today in Sardinia, and the hunter-gatherers, who have left the most ancestry in people today in northwest and northeastern and northern Europe, and that people today are a mixture of these two sources. So that was the model. But in 2012, we had in our laboratory another observation which was hard to reconcile with this model, and it was this. So what we found is we developed a statistical test, which we call a three-population test. And what we do is we take genetic data, variation data. So what we're looking at is hundreds of thousands of positions in the genome, single nucleotide polymorphisms, where some people have one type, say an adenine, and some people have another type, say a thymine. And we can score individuals for whether they have an adenine or a cytosine, or an adenine and a thymine, or something like this. And so what we did is we take a set of three populations, a test population that we're testing to see whether it's mixed, and then two possible source populations. And if the test population is mixed of lineages that are related sometime in the past to the source populations, the prediction is that the frequencies of these genetic variants in the test population will tend to be intermediate between the two source populations, because it's mixed. So the frequencies will tend to be intermediate. So we can test this, or the frequencies tend to be intermediate. At any one position, we can't measure that with any reliability, but if we average over hundreds of thousands of positions, we can obtain a very precise determination of whether the frequencies are on average intermediate in the test population relative to the source population. So we had this test in hand, and we ran this test on about 50 worldwide populations, present-day ones, and we looked for whether each population had evidence of mixture from any pair of the other two. So we ran our computer in a loop over the data, and what we found is that in northern Europeans, for example, present-day French people, there's a huge signal of mixture. One of them, the source populations, is Sardinians, southern Europeans, consistent with the idea that these are first farmers who have left the most ancestry in Sardinia, for example. And the other was not northern Europeans, but but it was Native Americans. So this was a really weird observation. And it definitely was Native Americans. It was not Siberians. It was not South Asians. It was not East Asians. The Native Americans gave a much stronger signal. And what we proposed is that what was actually going on is that there was in northern Eurasia, at some point in the past, more than 15,000 years ago, what we call a ghost population, a major population that lived in northern Eurasia in the past more than 15,000 years ago and contributed some of the ancestry of Native Americans living today, and that sometime later also contributed ancestry to Europeans. So this population doesn't exist in unmixed form anymore. It's been largely displaced since, explaining why people in Siberia today are not as good a source for European mixture as are Native Americans, but that this population existed in the past. So we call the population the ancient North Eurasians, and we think that they've contributed ancestry to the Americas and also to Northern Europeans. So that was the proposal of a ghost population statistically reconstructed based on present-day populations, no data from it. So what was very exciting to us was at the end of the next year, this ghost was found. It was found by a group, an ancient DNA group working in Denmark, led by S.K. Willerslev. And what they did is they sequenced DNA from a 24,000-year-old individual, a boy from the Lake Baikal region of east central Siberia. This individual was more closely related to Europeans even than Native Americans are, and was clearly the correct source population for the ancestry both in the Americas and in Europe. Today, the dark blue in this heat map shows that there's not much ancestry like that anymore, reflecting the fact that most of the indigenous people of Siberia today are the result of migrations that have brought people into this region from elsewhere more closely related to East Asians than the people were in Lake Baikal at that time. So this was very exciting to us that we could see the echoes of a ghost population writ in genetic data from present-day people, and that it could predict ancient DNA. But once the ancient DNA was in hand, everything became much more clear because it's much easier to look at it directly than to study it indirectly. So what we did, in work led by Joseph Lazaridis, working with Johannes Krause, then at the University of Tübingen, was to obtain high-quality data in 2014 from an early farmer from Europe about 7,000 years ago and a hunter-gatherer who lived about 8,000 years ago in Europe, and then to compare it to this ancient genome generated by the Danish group, and to try to build a model of population relationships. So I'm going to now digress for 2 minutes to try to tell you about how we look at our genetic data. So this is data from 777 present-day individuals from West Eurasia. So West Eurasia refers to this group of people who are genetically pretty similar to each other, including Europeans and Central Asians and Near Easterners. And these dots are colored by which population individuals are from, by self-identified ancestry, although the self-identified ancestry is not used in the mathematical technique used to display these points. So the nature of the data we have is single nucleotide polymorphism genotype array data. What this means is that we or others have looked at we have taken a genotyping chip which interrogates each individual at about 600,000 positions in the genome and scores that individual for what they have, adenine, adenine, adenosine, cytosine, or cytosine, cytosine, for example. So at each position, they will have 0, 1, or 2 copies of the mutation that chimpanzee doesn't have, for example. And there are about 600,000 rows corresponding to each of these positions, and then 777 columns corresponding to each of the individuals. We then multiply the matrix by itself and see how close each individual is to each other individual, and that's a measure of genetic similarity of all of these individuals. And then we run principal component analysis on that, and that separates the samples according to the most efficient way of separating that. So for example, the first principal component might be 0.3 times the value at position 1 minus 0.2 times the value at position 2 plus 0.4 times the value at position 3. And principal component 2 might be some other combination. And the mathematics of principal component analysis will efficiently separate samples in this way. So when we run this on present-day West Eurasians, we see a really dramatic pattern of 2 parallel gradients. All Europeans, almost without exception, fall in this gradient, with Sardinians at the bottom end and Northeast Europeans at the other end, and different points intermediate. And all Near Easterners, almost without exception, follow this gradient, with Armenians and people from the north of the Near East here, and people from the south of the Near East, like Bedouins and Arabians, over down here. So there are very few intermediate populations. The populations that are intermediate are populations with known or possibly known relatively recent contacts between the Near East and Europe, for example, in Greece or the islands of the Mediterranean, or Jewish populations. So what we did is we tried to look at the data and find a model of history based on genetic frequency similarity that is consistent with the data to the limit of our resolution. And I'm not going to describe the statistical tests used to determine that this model was consistent with the data, but we were able to show that this model was consistent with the data as far as we could tell. So we started with these ancient samples, this 24,000-year-old ancient North Eurasian, and this 8,000-year-old hunter-gatherer, and we found that they were consistent with descending from a common ancestral population, since separating earlier from East Asians and more distantly from sub-Saharan Africans. So far, so good. Native Americans are a mixture of these ancient North Eurasians. As I said before, they've contributed to Native Americans, but they're not all the ancestry. They also have ancestry from East Asians, and this was the main finding of that paper by this Danish group, that Native Americans today seem to be consistent with deriving from a very ancient mixture of two groups, one these ancient North Eurasians and another East Asians. There's always been an idea that Native Americans and East Asians are somehow sister groups that descend more distantly from West Eurasians, but in fact that's not true. It's more complicated, and this is a potential resolution of that. The first farmers of Europe are a mixture of an ancient group related to the hunter-gatherers of Europe and another very distant population that's actually a new ghost population we predict from the data. We call them the basal Eurasians, and we've gotten closer to finding them now, but we haven't quite found them in terms of ancient DNA. It's a very interesting lineage because it's separated from the ancestors of West Eurasians and East Eurasians before they separated from each other. And people in Europe today can only be modeled as having mixture from all three of these sources. So we have a three-way mixture, not a two-way mixture. So the mixture proportions vary depending on which West European population they are, but almost everybody has ancestry from all three sources, from the hunter-gatherers, from the farmers, and from the ancient North Eurasians. So what actually happened is the ancient DNA had shown that the farmers come into Europe, displace the hunter-gatherers, or mix with them, but all the ancient DNA that had been studied up to this time only showed farmer and hunter-gatherer ancestry. But today, Europeans all have this ancient North Eurasian ancestry. So something must have happened to bring this ancient North Eurasian ancestry in. So that was the next thing we did. We tried to actually find when the ancient North Eurasian ancestry entered Europe. And so the way we started this is in a paper based on mitochondrial DNA ancient sequences, which looked at DNA from about 300 mitochondrial sequences from 9 successive archaeological cultures defined based on their pottery, dating from about 7,500 years ago to about 3,500 years ago. And they start with the first farmers or even the hunter-gatherers and continue until the early Bronze Age, when people start using new metals much more widely in Europe. So what we developed is a test of continuity. Is it possible that this population, the second population, is consistent with descending directly from the first without subsequent mixture, and so on? And so we had a grid where we test for continuity between each of these successive populations, the hunter-gatherers, the first farmers, slightly later farmers, and so on. And there's a colored box here if there's significant evidence of difference. And what you see is a line of very dark-colored boxes showing that the hunter-gatherers are discontinuous with all the subsequent farmer populations. This is, again, the evidence of hunter-gatherers replacing farmers replacing hunter-gatherers. It's very dramatic. But after the first farmers arrive, about 7,500 years ago in this part of Europe, there's continuity for the next 2,500 to 3,000 years. There's no very strong evidence of new major input. But then, bang, 4,500 years ago, there's discontinuity again, beginning with a culture called the corded ware culture, and ever afterward, there's discontinuity with the previous time. So this is a hint of what happened. The way we explored this further is we collected genome-wide data on a large subset of the same individuals, plus additional individuals, many of them from Central Europe, from Germany, many of them from Spain, and a whole important series of samples from the Samara region of Russia, which is far Eastern Europe, and it dates to just before the corded ware period. So now I'm going to actually give you a sort of still movie of what happens over time in Europe. This is the same picture I showed you before, principal component analysis. Remember, here's the Near East, here's Europe. I'm now going to gray out these dots so that you can focus on the ancient samples. So here's the Near East, here's Europe, and the question is, how do the ancient samples fall relative to present-day populations over the time? So if you look at the hunter-gatherers of Europe before farmers arrive, hunter-gatherers fall beyond Europe in the direction of European differentiation from the Near East. What this is saying is that all of these hunter-gatherers from Sweden here, from Western Europe here, and Spain and Luxembourg are from a population that doesn't exist in Europe in unmixed form before anymore, but that Europeans today have ancestry from them because they are moved and shifted in that direction relative to the Near East, which you can actually show formally using modeling analysis. In the far east of Europe, you actually have populations like this on the West Eurasian steppe, which we call Eastern hunter-gatherers, which are pointing at the high end of this gradient, similar to that ancient North Eurasian, and we think they carried their later population that carry this ancestry. And so Europeans today are arrayed on a gradient of different proximity to these ancient North Eurasians. So you can see both gradients represented here, the hunter-gatherers responsible for this shift and the ancient North Eurasians represented for this gradient, by this gradient. So now comes the first farmers, and there's a large cluster of samples we have here from all over Europe that are similar genetically to present-day Sardinians, as we knew already, but present-day Europeans, except for Sardinians, position at a place that is not yet in this represented by any ancient samples. At the same time, what happens after that is the slightly later farmers shift slightly toward the hunter-gatherers. What's happening here is that the farmers who initially arrived were socially segregated from the hunter-gatherers, but over the next 2,000 years, they began to mix with them. So those social barriers broke down, and we can see that happening in Spain, and we can see that happening in Sweden, and we can see that happening in Germany and Hungary. It's very exciting to see this happening in parallel everywhere. Simultaneous with this, in far eastern Europe, another mixture occurs. These eastern hunter-gatherers mix with people from the northern Near East, from places like Armenia or Iran, and form a mixed population here analogous to these mixed populations. But at that time, 5,000 years ago, you still don't have populations like British people today, like Northeast Europeans, even like most Southern Europeans. So when does that happen? Well, it happens 4,500 years ago with the corded-ware and the succeeding groups. Suddenly, these groups, like this and this, mix together, and we can show, using formal statistical techniques, that it's very difficult to get this mixture to happen without groups very closely related to these and very closely related to these. These people are what are called the Yamnaya. They are steppe pastoralists. They're herding sheep on the steppe. They took their sheep out onto the steppe for the first time after beginning to use wheeled vehicles, carts and wheels and horses, which allowed them to move out in mobile homes onto the steppe. And they took advantage of this landscape, which was unexploited before, and expanded very dramatically. There had been a controversy about how they affected Europe, and what the genetic data shows is that they actually had a major genetic impact on Europe at this time. So just summarizing the population changes after the advent of agriculture in Europe, 7,000 or 8,500 years ago, the first farmers land in the Balkans and Greece, coming from an ultimate origin in the Near East. We have ancient DNA now from Turkey, and it's a good match to that, which matches a lot of archaeological hypotheses about where they came from. And if you measure the proportion of Near Eastern ancestry in farmers at this time, it's approximately 100% measured by this orange, the farmer ancestry. In the next 2,000 years, the farmers and the hunter-gatherers mix, as I mentioned before, as these social barriers break down, but there is still no ancestry related to the steppe, and that's ubiquitous in Europe today. The next thing that happens 4,500 years ago is populations appear that are more than half steppe ancestry related to these Yamnaya, and they come from the steppe. And people in Europe today, especially Northern Europeans, have many of them at least have half ancestry. In many European populations, this is now the dominant ancestry contribution to Europeans. Now you may ask, before I said that the ancient North Eurasians are only a quarter or eighth of the ancestry of present-day Europeans, but now the Yamnaya are a much larger proportion. What's going on is the ancient North Eurasians are a tracer die. They actually weren't the whole ancestry of the Yamnaya. The Yamnaya only had perhaps a quarter of ancestry from this population, and so they put a minimum on the proportion of ancestry coming from the east, but the Yamnaya actually brought a larger proportion of ancestry in, and this is actually probably the single most important contributor, or along with early farmers, to Europeans today, these people. This is not only our work, another group publishing at the same time as us, this Danish group that I mentioned before, published a parallel paper and came to similar conclusions based on their independent data set of a dramatic turnover in Europe at this time related to the Yamnaya. So now I'm going to expand a little bit more broadly in West Eurasia and talk about the broader West Eurasian context of what happened in Europe. So Europe is a peninsula of Eurasia, which is an interesting place, but it's a broader part of this broad region where today, genetically, people are very similar. As I mentioned at the beginning, people in West Eurasia today are genetically strikingly similar. So was it always that way? So we just published a paper a couple of months ago where we got DNA from 44 ancient samples from the ancient Near East, ranging from 14,000 years ago to 4,000 years ago, from Iran, from Israel and Jordan, from Armenia, and they include people from before the transition to farming, from just after the transition to farming. They include people from the east of the region, and they include people from the west of the region, and they include people moving into the Metal Ages, into the Copper Age, into the Bronze Age over this period. So what we did, so I'm going to just digress a little bit, and what made this ancient DNA work in the Near East possible, and I think what's going to make work in Africa and in South Asia possible too, is this innovation of the petrous bone. So the petrous bone is a piece of your temporal bone. It's the part of your bone, your temporal bone, that is around the inner ear region, and it has been realized in the last year or so, year or two, that the petrous bone contains about 100 times higher density of DNA and skeletal remains than any other part of the body that people have looked at, any part of the skeleton that people have looked at. And so ancient DNA scientists have moved toward preferentially trying to sample from the petrous bone where possible, because it's so much richer a source of DNA than any other part of the skeleton people have identified. And that, combined with this enrichment technique I mentioned, has increased success rates a lot. The Near East was a very difficult place for ancient DNA because heat is bad for DNA preservation. And so, but using the petrous bone and enrichment, we have been able to get working DNA, and others have also had successes in other really exciting contexts. What we do is we take these petrous bones, we use a dental sandblaster to remove the part of the petrous bone containing the cochlea, which is the part of contains a lot of the apparatus of hearing. Here's an isolated cochlea. We grind it, we mill it. In a miller, we obtain powder, and then we subject that to the later stages of ancient DNA processing. So here's an updated version of the picture I gave you before, but now with more samples, and there was not samples here before because we now have ancient samples from the Near East across this whole gradient, people at the bottom of the gradient and people at the top of the gradient. And what we find is that ancient Iranians are at the top of the gradient, and ancient people from the southern Levant, from Israel and Jordan, are at the bottom of the gradient. So they too have different proportions of ancestry. And in fact, if you measure the level of differentiation between the different groups in West Eurasia from 10,000 or so years ago that we now have data from, it's absolutely huge. So if you use FST, which is a measure of how different populations are, it's related to the squared frequency difference between pairs of populations. All these four populations at the extremes of the quadrant, the first farmers, the hunter-gatherers of Western Europe, the Iranians, and the Levantine, the first farmers of the western part of the Near East, they all are as different from each other as Europeans and East Asians. So West Eurasia, 10,000 years ago, has four groups that are as distinctive from each other as Europeans and East Asians. And today, there's a very little difference. So what happened? We can track it through time with our ancient DNA data. So at this time in the past, 10,000 years ago, the difference is as large as Europeans and East Asians. It then drops so that by 4,000 or 5,000 years ago, it's hit levels seen today. So what's happened? Well, what's happened is this. 10,000 or so years ago, you have these four approximate sources of ancestry in Western Eurasia: Western European hunter-gatherers and groups related to them, Eastern European hunter-gatherers and groups related to them, first farmers from the Western Near East, first farmers from the Eastern Near East, and then later populations across the region arise from mixtures of these groups. They mix dramatically, and the reason differentiation amongst the groups today is so low is because of mixtures of these groups. It's not because one group has replaced the others, but because they've all mixed with each other. So the Near Eastern farming expansion and its after effects homogenized West Eurasians from these four or so sources at the beginning to a region of broad homogeneity, which looks so similar today, and a lot of people sort of imagine as ancient, but in fact is a very recent phenomenon. Hypothesis is that East Asian and African agricultural expansions may have similarly homogenized those regions, and in a similar way, those categories we have in our heads may actually be pretty artificial and not very long-standing categories, but there's no ancient DNA that's comparable to what's in Europe yet. So in the final part of my talk, I wanted to talk about the insights about biology that are coming from ancient DNA and studies of modern DNA that related to, and that I think are exciting. So I'm going to first introduce the idea of a selective sweep. So a selective sweep is when a single mutation occurs in a person that's advantageous, that might allow you to digest cow's milk into adulthood, for example, if cow's milk is available as a source of nutrition. It occurs in one person, and then the idea is that it rises to high frequency in a population under the pressure of natural selection. That's what's called a selective sweep, and it's really easy to detect using genetic patterns because what happens around a selective sweep is everybody descends from that individual who had that mutation pretty recently in time, and that leaves a scar in the genome in the pattern of variation that's easy to detect. So here's an example. If you screen across this chromosome 2 and look for places in the genome that have strong reduction in variation measured in one way or another, you'll see a big peak, for example, at this region that allows you to digest cow's milk into adulthood, the lactase gene region. But actually, Molly Jaworski and colleagues have shown that, in fact, selective sweeps actually are relatively rare in human evolution. There's a lot of evidence for natural selection, but selective sweeps have not driven most of it. Instead, most of it seems likely to be selection on pre-existing variation in the population that suddenly, under a new environmental condition, becomes advantageous. And that won't leave a scar in the genome. So in a way, when we do these scans for selection, we're only finding what we can easily find, the low-hanging fruit. We're not actually finding most of what matters. So what we actually did with our ancient DNA data is we took advantage of the fact that ancient DNA data gives you time series data. It's actually the first, we understand as experimentalists the power of being able to do an experiment and watch the experiment develop over time, but in humans, until ancient DNA, we've had to be stuck with people in the present day, and we haven't been able to watch how populations evolve into each other over time. So instead, what we've actually tried to do, what we can do with ancient DNA is watch, for example, European populations and how they change over time. So what we did is we took this model that we had fitted to the data of Europeans today being a mixture of 3 highly different ancestral populations, and then we took each position in the genome and we asked, does that position in present-day Europeans from different populations have the frequency that you would expect based on that mixture of those 3 ancestral populations that we fit based on the whole genome? So we run this across the genome, and we carry out what's essentially a genome-wide association study, but not for affecting diabetes or not, but whether it's changed in frequency more than you'd expect by chance. And we find at least 12 genome-wide significant locations with the data that we had available at the time that we did this scan. Some of them are known variations, and some of them are ones that have not been identified before. I'll give you a few examples. So this is lactase persistence. This is a mutation that allows you to digest milk into adulthood. In all of the ancient samples, ranging from about 8,000 to 4,000 years ago, it's very rare. And today in Northern Europeans, it's very common. And so you can see why there's very strong evidence that it's changed in frequency. And really, it only begins to be seen in Europe about 4,000 years ago. Similarly, for light skin is lower in frequency in all of these ancient populations that include the sources of Northern Europeans than it is in Northern Europeans today, and also even then in Southern Europeans. Now, light skin, the main variants affecting light skin seem to have come in, for the most part, from the ancient Near East with the farmers. Blue eye color is really from the indigenous hunter-gatherers of Europe, and there seems to have been repeated types of selection on this variant. But for us, this was not the most exciting type of work. For me, the most exciting type of work was the genetic our ability to look with this data at selection for complex traits, for things that are not underpinned by a single gene, but many genes. So where we had our most success in this area, and what I'm most excited about for the future, is in the case of height. So height is a trait that's been studied in hundreds of thousands of people now in medical genome-wide association studies, and so we've annotated as a community millions of positions in the genome for how they affect people's height. And so we have this annotation that we can then correlate to changes in frequency over time. So there's clearly a genetic underpinning to height in Europe. While height's grown over time across Europe, the order in which populations have ordered by height has remained about the same, with relatively small stature in Portugal, relatively high stature in Netherlands and Croatia, for example, and that's changed over time. That's remained constant over time. In 2000, Lengo Allen and colleagues published a very important paper where they found 180 independent positions in the genome that affect people's stature. A later paper by Joel Hirschhorn and colleagues found that there's been selection for different stature in Northern and Southern Europeans. They found consistent evidence that the variants affecting taller stature, larger stature, were more common in Northern Europeans than in Southern Europeans, and that this was too much to be explained by random fluctuation. So there must have been selection for either taller stature in Northern Europeans or smaller stature in Southern Europeans, but they didn't know which. So here's where the ancient DNA came in. So we had ancient DNA from each of these ancient populations, and we could score them for whether they had what their predicted height was based on the genetic variants that from present-day genome-wide association studies know affect height. So the first farmers are over here, and then there is as populations move into the middle of the first farming period, there emerges a difference between Northern European farmers and Southern European farmers, and there's a statistically significant drop in genetically predicted stature in Southern Europeans. So what this means is that the reduced stature in Southern Europe is actually due and driven in large part by selection to reduce stature. It's not selection to increase stature in Northern Europe. And there's a separate effect in the steppe where there seems to have been selection to probably increase stature in the steppe, and these people then come into Europe 4,500 years ago. So that's what's going on. That's what's explaining it. Now, it's not clear why taller or shorter stature would have been favored in certain environments, but it clearly was, and we can determine the directionality of it. So I think that this is a really exciting way to interrogate how traits evolve over time. And I think it combines the power of genome-wide association studies, which have now been carried out for many traits, diabetes and heart disease and many cancers and autoimmune diseases, as well as height and other traits, which have resulted in annotations of the genome of the effects of variants. Often they're very, very minor effects, but we can correlate them to changes in frequency over time to see whether there's been selection in one way or another for certain traits. Height is the best understood trait, but the paper by Jonathan Pritchard's group that's on a preprint server and that I hope will be published soon, finds evolution not just of height, which is the strongest signal, but of infant head circumference and insulin levels, age of puberty, especially in females, and you can actually see each of these traits has been selected in Britain, which is where the study is based, in the last 2,000 years, and in the same type of way by correlating changes in frequency inferred from genetic data with the annotations from genome-wide association studies. So I want to finally conclude by asking the question, if you go back in time, how did distinctively modern human traits appear? So I'm going to give you a little background on this. The first anatomically modern humans, people with skeletons similar to ours, date to almost 200,000 years ago in East Africa. And the archaeological studies of these people, of their stone tools they made, the ornaments they left behind, indicate that they're not likely to have been any more sophisticated behaviorally than their cousins in Europe, for example, Neanderthals, or elsewhere in Asia, or elsewhere in Africa. That is, these people were anatomically like us, but they were behaviorally just like Neanderthals. They made similar tools. And it took 150,000 years for these people to actually become behaviorally much more like us. Archaeologically, there's documented about 50,000 years ago a quickening of archaeological change where much more complex tools start to be made, including bone tools, not just stone tools that are left behind, clear evidence of people sewing, clear evidence of a lot of ornamentation, cave art, figurines, deliberate burials. All of these perhaps had occurred sometime before, but much more intense, and changes every few thousand years rather than every tens of thousands of years in styles of stone tool production. So archaeologists seeing this thought, what you're actually seeing here is evidence of a great change in the human population, And perhaps it's even a biological change. Maybe there's been a genetic switch, a change in maybe a neurological gene that wouldn't have left an imprint in the brain case, but rose to high frequency, and it allowed complex behavior or maybe conceptual language, and it permitted one population to expand. That population expanded outside of Africa after about 50,000 years ago, largely displacing all the people who were there. It probably expanded within Africa too, mixing with or displacing most of the populations that were there. That is the idea that was very exciting. And this idea of a genetic change that might somehow explain these quickenings, these great changes, what makes us distinctive from Neanderthals and our relatives, has been invoked by a lot of people, including me, to justify searches for evolutionary studies, or we're going to find the key to what makes us special. And we're going to find, by sequencing the chimpanzee genome or sequencing the Neanderthal genome, we're going to look at the differences between us and chimpanzees. We're going to read it like a book, and we're going to figure out what makes us special. And the problem is we still haven't figured out anything that's very meaningful about what makes us special based on comparing to these genomes, even though they've resulted in such amazing successes in other ways. So what I want to conclude on is some work that speaks to some of these questions. It's this paper that Francis referred to just before, which is a study we just published of 300 high-quality genome sequences from 142 diverse populations from the places shown in this map. It contains a lot of variation that's not in public resources, especially from populations that have not been sampled before, for example, from populations from Africa or from New Guinea and elsewhere. So the first thing that we can do is we can line up the genome sequence that each individual inherited from their mother and each individual inherited from their father, count the number of differences between them, and since differences accumulate over time, they provide a measure of time. And so we can ask, how long has it been since and you can do this, compare two populations to each other as well. And so what we've done is we've used the technique developed by another group for estimating population separation. And for many pairs of population, much of the separation happened before 50,000 years ago, which is when this great change occurs. So the first thing to keep in mind is that these big changes that have occurred in the archaeology happened before the separation of a lot of present-day populations. And so if you believe that there was a big change, it must have occurred more in the ancestry of some human populations than in others. But of course, all populations today are capable of fully complex language and so on, so this is perplexing. The second result is that we actually didn't find any places in the genome that are plausibly contain a genetic switch. We used the same technique to estimate at each place in the genome how far back it is since you share a common ancestor by just counting density of mutations. And so this is a result on a chromosome for person 1 is how far back in time do the two chromosomes, the two copies of the genome that an individual carry, share a common ancestor. And so we just do this for many, many individuals, and we see over everybody today, how far back in time do we have to go for everybody in the population to share a common ancestor? And the answer is that there's really nowhere in the genome where everybody shares a common ancestor more recently than 400,000 years ago. Now, we haven't gotten every last section of the genome, but most of it we've covered. And if you believe this genetic switch idea, you must believe that there's a genetic variant that rose order of 100,000 years ago, rose high frequency by about 50,000 years ago, and that then was ancestral to almost everybody today. But that's not what you see at all. There's nothing even plausibly close to that. And so the conclusion of this part of the talk is that I think that, for me, it's suggesting that there's not likely to be simple explanations for behavior, evolution of these behaviorally complex traits that are evident in the archaeological record, and maybe you might generalize more broadly and wonder if there might not be simple explanations for other evolution of human traits. We don't think there's any evidence of there having been one or a few enabling mutations that suddenly appeared amongst our ancestor and allowed them to think in profoundly different ways, that it was somehow a population in what we call a mutation-limited state, which couldn't do things before, and then suddenly an amazing mutation happened and allowed people to do things, because we don't see any evidence of mutation that could have been like that. I think that distinctively human behavior, because of this, it seems more likely to be a complex trait, not like skin color or lactase persistence, and that human populations 50,000 years ago already had within them the mutations necessary to do everything we do. And it seems in this context that maybe environmental and cultural changes, which perhaps precipitated frequency changes of those existing variations, so genetics could have contributed, are likely to have been drivers of great changes that occurred. And it's an interesting case here where geneticists often search for examples where genetics is the explanation, but here in a sort of paradoxical way, genetic data are showing that there are unlikely to be sort of reductionist genetic simple answers to what makes us special or different. But I actually think that that's an opportunity in a way because I actually think that the interesting insights will come from trying to embrace the complexity rather than to come up with a simple explanation. So that's the end of my talk, and I think there's questions. Time for a few questions.
Thanks, David. That was fascinating indeed, and I suspect people do have questions. Please use the microphone so that people listening on the video can also hear the question. Please start right here.
Yeah, I'm wondering about disease correlations and with your patterns of the 3 or 4 European backgrounds. Is there any linkage to any particular diseases? In other words, can you say that some diseases, genetic diseases, originated in those different populations?
The question is whether certain diseases are likely to have originated in one population or the other. We, in fact, try to look at the Genome-wide association catalogs and correlate them to different contributions in one way or the other, and we haven't really obtained any adequate results in that way except for height yet. I think it's in part due to the limitation of the ancient DNA data we had available at the time. This is driven by sample size. So I think in order to obtain better insight, what we want to do is increase the sample size, but that's happening very rapidly now with the increases in the quality of the ancient DNA data that are becoming available. So I think that combining with the ancient DNA data with the genome-wide association studies that are for these different diseases, we might be able to partition diseases in this way.
David, great talk. Just clarification at a technical level. At the beginning of the talk, I got the impression that the enrichment methods you were using, you were sampling actually a very small fraction of the genome, but getting lots of coverage of it. Now, is that always, when you refer to when you use the phrase genome-wide, does that mean just that enrichment, or do you literally mean attempting to get the entire genome? And then that coming late in the talk, when you talked about your 300 genomes, again, what fraction of the genome is being sampled?
Right. So the genome-wide data, what we're using in our laboratory is an enrichment technique that doesn't sequence the whole genome for most samples. We sometimes do sequence the whole genome, but we enrich basically about 1.2 million positions that are of interest. For a subset of the samples, it's as low as only 5% of positions covered. But in fact, many of the samples, the median coverage might be 0.3, and many samples are heavily covered at those positions. But it's not every position in the genome, and that's the secret that we've used to being able to study very large numbers of samples. There is a possible thing one could do, which would be to shotgun deep sequence a lot of these samples that are amenable to it because they have high percentages of human DNA. That is not what we have done on these samples so far, but it's possible and an exciting thing to do. The last question you asked was about the Simon's Genome Diversity Project, the 300 genomes, and that's present-day genomes sequenced to high quality every position in the genome that we could sequence.
And then just one last question. When you showed the Moore's Law growth of ancient DNA sequences in general from many investigators, in general, are most of those sequences like yours just sampling some small percent of the genome, or are many of those a larger fraction?
So our laboratory has produced more than half of the sequences that have been published to date. So most of them are published, are from that technique, but a substantial fraction of our shotgun sequence data.
The grants from Princeton have done studies over several decades now looking at Darwin's finches and looking at beaks, and have noticed that there's an evolution of beak size based on environmental impact. And I'm just curious to know if you look at the particular points in time where you see the different groupings, do they merge, and is there sort of a blending and at times of geological stress? And then another question I had to deal with whether or not, like you mentioned, the lactose and ability to digest milk. Are there gene drivers associated with that so that it becomes more and more prevalent within a population?
So, yeah, so I think, again, we're at the point where we don't have enough ancient DNA yet. Most of our ancient DNA is from a relatively narrow time slice that I reported on between about 7,000 years ago and about 4,000 years ago. And so in order to really document in detail how the changes in frequency occur over time, I would want to fill in the more recent period and allow one to track exactly when the changes occur. Really, in some approximation, we have two time points. We have the first farmers and their immediate successors in Europe and the present. And so that's 2 points, and we can't really do more than draw a line connecting those 2 points. But with more data, we can actually fill in that line and understand when the critical changes happen. With lactase persistence, you can actually see in our data when it first arises. You can see the first occurrences in substantial frequency about 4,000 or 5,000 years ago. And your other question was?
Had to do with gene drivers. I'm just wondering, you have a mutation, and then it seems to become more prevalent in a population, such as lactose tolerance. And I'm wondering, is there something that's driving that prevalence?
I don't, my guess, I don't know, but I think what we know, I think that what we generally assume is that the increase in frequency of lactose persistence is driven by the importance of milk in the diet, and that's very advantageous to be able to digest it into adulthood, and so people have more offspring if they are able to do that. So it's just natural selection. I don't think that we have any evidence of anything other than that, although that would be interesting, but I'm not sure I'm answering your question.
Yeah, no, you answered it. But as you mentioned, that you're looking at a specific gene, and you're not looking at what's may be associated with that gene. Is that correct?
That's right. That's right. There's a lot to do.
Over here.
You mentioned this narrow time slice of samples you're looking at. Is there any possibility you can compare your samples, and in particular the ghost populations you anticipate, to existing older ancient DNA, in particular the Neanderthal genome and the Denisovans, to see if those ghost populations in any possible way are related to the more ancient data?
Yeah, I've done a lot of work. Most of my work in ancient DNA has, in fact, been on Denisovans, Neanderthals, and the relationships between these groups. One thing that we can see, and that's actually in this work on the Near East, is we can actually see that there's been well, actually in another paper we published a few months ago, which was on Upper Paleolithic Europe, so Europeans, 51 samples between 45,000 and 7,000 years ago, is that there's actually been a decrease in Neanderthal ancestry over time in Europe. So that in the first European modern humans that we have data from, or Eurasian modern humans, it's about 5%, and it decreases to about 2% today. So there was more Neanderthal ancestry in the past. And it's actually particularly interesting because these populations are directly ancestral to some of the later populations. So we think there's been natural selection to remove Neanderthal ancestry. It's slightly disadvantageous, but we can actually tell very accurately whether the proportion of ancestry in of an individual genome that comes from Neanderthals and Denisovans, and none of these groups have extremely large amounts, except for one sample we found which had about 10%, and we found that their 4th to 6th degree relative ancestor was a Neanderthal. So we were very lucky we found a 4,000-year-old and 40,000-year-old individual like this. But most of these samples are much less and not particularly unusual for their time.
David, let me ask you about what you perceive as the maturity of this very rapidly moving field. Amazing observations that have come forward in just the last few years. But what do you think is going to happen in the next half-decade? Are there many mysteries yet to be revealed, or is the big picture now pretty much taking shape?
Thank you. I think that we're really just at the beginning. We've really... There are so many things to do, so I can just rattle off a series of things that one could in principle do. We could increase sample size tremendously, and now that's very easy, and we can actually really study natural selection with really high statistical power. The study I told you about was just a proof of principle, but there's really the possibility of really doing this with extraordinary accuracy, of doing scans of what natural selection landscape was like 10,000 years ago and comparing it to the present. Has selection changed in its nature? Another possibility is to study what happened after the Bronze Age in Europe, for example, what transformations happened since these people arose. Europe is a tiny part of the world. One could study the Americas, one could study East Asia, one can study South Asia, one can study Africa, many parts of Africa, and there's all of these places to study, and I did not mention many places. There's also things that one can do with ancient DNA which will answer questions that have not been answered. we can probably, with ancient DNA, just like we can of present-day populations, use DNA to estimate the census size of populations. You can see genetically in people today that Han Chinese have a very large census size because, on average, they have very few close relatives. Any two random Han Chinese, you can see this in the data. So if we can get substantial sample sizes of ancient people, we can estimate population size in the past, something we've never done, and that's so important for economics, for health even. Another thing that's very interesting is that this big arrival of people from the steppe, how did the people from the steppe herding sheep come into Europe where they were densely populated by farmers? How did they make an impact where people who already had a more efficient way of living can make an impact? There was a study by this Danish group that I mentioned to you that studied the genome sequences of these samples at the time of this impact, and they found that in these 4,000 to 5,000-year-old individuals from across the steppe, there is plague bacillus, there is the Black Death plague, and which some people thought had only jumped into the population maybe in the 14th century or maybe with the Justinian plague in the 7th century, but here it is 5,000 years before. It doesn't have the virulence factors needed for bubonic plague, but it does for pneumonic plague. 5 or 10% of the samples that they studied had plague in their teeth at the time they died, which means they probably died of it. So you have plague endemic on the steppe, and one possibility that these people suggested, which is an exciting possibility but shows the potential power, it's certainly not proven, is that maybe Europe 5,000 years ago was like Native Americans were 500 years ago. Maybe it was a naive, genetically, immunologically naive population that was then suddenly exposed to diseases of the steppe, and that there's big diseases, great diseases that swept across the steppe, Europe at this time, and made it possible for people to move in who otherwise would not have had the demographic niche. So there's so many things to do, I think.
I think he's right. There's a lot of work to do here. If you want to continue this conversation with David, please join us for coffee and cookies over in the library. And meanwhile, let's thank our speaker again.